I'm Leigh Winowiecki. I'm a soil scientist, and today I'm going to talk about advancements in soil and rangeland health monitoring alongside my co-authors. So to get us started, probably no surprise to this community, but we did a review of 6,000 publications aligned with the UN Decade on Ecosystem Restoration. And this graph here shows the ecosystems, forests, grasslands, drylands. And on the right-hand side, it talks about where most of the publications were about. So finance, political will, scientific research. The thicker the, so only 3% of the publications were from drylands and only 6% were in grasslands. The thicker the line means there's more, the bigger data gap. So I think there's a real obvious need to fill these knowledge and data gaps in rangelands. And these data can be used to prioritize interventions, investments, and policies. So today I'm going to talk about the need for systematic monitoring of rangeland health. This will help us assess the impacts of management, establish robust biophysical baselines, including CEF for any carbon projects, provide consistent protocols to track changes over time, and to really understand what works where and for whom. So today I'm going to talk about the LDSF monitoring framework. So it's called the Land Degradation Surveillance Framework, LDSF. This is a photo of the team in northern Kenya in Kalama. I should have asked our Dalmas to guess where this was from. And it was designed in 2025 to provide a biophysical baseline. And we have now been implementing this for 20 years. So take out your phone and scan the QR code. This is a really cool blog we did on kind of the evolution of the LDSF. Each green dot here is actually a 100 square kilometer LDSF site. So yes, I'm glad our speaker is going to talk about North America because we are focusing on the global tropics, as you can see from the map. So applying a consistent framework across landscapes allows us to really do comparisons and also produce accurate maps of soil health. So we have a soil sampling procedure where we collect soil samples, composite at the plot level. We also do vegetation measurements. So this is a photo of measuring of trees, and I hope you see the elephants in the back. This is Lumo Conservancy in eastern Kenya. We also look at water infiltration. So as our previous speaker said, that these areas are often prone to prolonged drought. So really understanding what happens when water enters the soil. Does it enter the soil or does it run off? And how quickly does it flow through the soil? We also have a rangeland health module where each plot has a transect where we look at different vegetation species. So I don't know if we have grass lovers here, but we can dive into some of the species where we do grass, forb, and woody vegetation along the transects. And a really important component of this is it's also collected using electronic data entry. So these data automatically are uploaded to the global database, which is really aids in data analysis, data management, data storage, data sharing. So what do we do with these data? How can we make sense of these data? Well, one is we can compare soil organic carbon across sites, across management, across restoration. So we collect all of our soil samples using an auger here. And let's look at some graphs. So on the y-axis are a number of sites in East Africa, Uganda, Kenya. Each site was how long? Big, 100 square kilometers. So that's 10 kilometers by 10 kilometers. And they all have 160 topsoil samples and 160 subsoil samples. So these box plots show the topsoil carbon and the subsoil carbon. Now, why am I showing box plots on a webinar? It is because they're so telling. So the wider the box plot, the more variability there is. The more narrow the box plot, the less variability. So just looking at this now, as we have for a couple of minutes, we can see that sites that have a wider box plot like Mara and Lewa also have higher carbon. The sites that have more narrow box plots, like Lokiriaman in Turkana and in Balambala and the Upper Tana, have lower carbon values. And this is also very worrying because when you start to have less variation across a 100 square kilometer site, it starts to indicate that we may be approaching some tipping points and some thresholds of degradation. We can also produce maps, so pretty maps, right? Not only are they pretty, they're based on field data, so they're accurate. So this is a ranch in central Kenya, Laikipia. On the left is a map of soil organic carbon. The darker the color, the higher the carbon. We also produce maps of soil erosion. So the yellow and red are hotspots of erosion prevalence. So we can also combine these data to create restoration priority areas. So that's the resulting map. So gray area is a degraded area with semi-low carbon and semi-high erosion, where moderate efforts for restoration would be needed. The red is highly degraded with very low carbon, very high erosion, and it will require more intervention. And for those of you who aren't familiar with the Laikipia landscape, here are some photos and a photo of the Lintonia grass species there. Oh, speaking of grasses, why do we think that there is so much carbon in rangelands? It's because of these beautiful roots of diversity and diversity of root architecture that exploit the soil profile and actually provide organic matter to the soil below ground. So in the LDSF, in the rangeland module, we actually look at these grass species. And for those of you who know me, know that I love grasses. I love identifying grasses, and we have many programs engaging youth in monitoring so that this local knowledge, even the names of these grasses, are revitalized across the community. So I'm just giving you an example of two different conservancies here in Laikipia. One has 39 grass species, with the most common species is this Themeta triandra. So Seth, you saw this a lot in the Mara. And then second is the cynodon. This grass on the right, the most common species was Eragrosa superba, which is photographed here, and it doesn't even have the Themeta triandra in the top grass species. So why is this important? When we're designing restoration, we need to consider the right grass for the right place. So in Omalo, the grass species composition is different than in Loisaba. So we are building one of the largest systematic databases of grasses, annual, perennial, and forbs and woody, and really building this database. Does anyone know how many grass species there are in Kenya? There's suspicion that there's several hundred, but how many grass species are commercially available for restoration? 3. So there's a lot we can do in terms of grass nurseries and revitalizing some of these less common species. This is a really cool project we had with the Laikipia Conservancies Association and the Nature Conservancy, where they implemented LDSF clusters, so 10 LDSF plots per cluster across 27 conservancies. That's the map on the upper left. Even if you can't read the graph here in the center, but you see on the y-axis, there's a list of the conservancies. And the value here is the saturated hydraulic conductivity, so infiltration capacity. And just look at the variability across conservancies. At the top, you have Mugia, it had the highest infiltration capacity compared to Old Mysore or Pingwan. So it's really important that we do systematic monitoring so we can make these comparisons. On the bottom right is actually a graph of the conservancies again, but then how many perennial grass species did they have? So very interesting, Solio only had 14 perennial grass species. Mukatan, 44 perennial grass species were recorded in the survey. So just going back to that point of we need to really understand the species composition in order to prioritize and intervene. And I can't leave here without talking about soil biodiversity. So this is an amazing project we have with SLU in Sweden and Restore for More project. And arbuscular mycorrhizal spores are key indicators of soil health. Again, on the x-axis, we have 4 rangeland sites. Those are the names, all the same size. But my goodness, can you see that the Lokiriyama site has the lowest number of AMF spores? Now, who remembers which site had the lowest carbon as well? It's also Lokiriyama and Turkana. So we use AMF spores as an indicator, both in the number of spores, but the diversity. So Luglicia Mwangi is doing her publication and her PhD on eDNA, AMF spores, and even soil acoustics in these rangeland systems. I only have 2 more slides, so I just wanted to highlight that luckily there is interest in developing more consistent monitoring frameworks or implementing them like the LDSF. So this is the Stellar project. So Boni is from ILRI, and this project is led by ILRI with IUCN and a number of partners. And our role at ICRAF is to develop a rangeland monitoring framework to support the upcoming rangeland standard. So this is very exciting so that we can actually track the impact on KPIs. And again, please scan your QR code here. The Coalition of Action for Soil Health was founded in 2021 at the UN Food Systems Summit. And we have Colleen Knox, the Director of Communication, on the call, and she can put in some links in the chat. And we have a campaign on the IYRP, and we would love to get your contributions, share any of your materials on this website, get your quotes, and shout all about rangelands. And of course, even in our annual report, we had a foreword from youth pastoralists. So we're really doing as much as we can to raise awareness, bring evidence and data to life, and elevate the voices of marginal groups, of youth, of women, of pastoralists, of really the whole community. So with that, my key messages are that there is an urgency to invest in rangeland restoration and soil health. There's a need to fill these knowledge gaps on the impact of interventions, but we do have the tools to monitor rangeland health, including soil organic carbon. I didn't get a chance to talk about it today, but we have a citizen science tool called the Regreening app that actually has a rangeland module. It's in 7 different languages and 400,000 users. So with that, I just want to say thank you so much, everyone. I'm really happy to be here, and thanks, Seth, for your leadership in this webinar, and happy Earth Day.